So I'm Aurélien Luciani. I will be talking about the UniProt website and the development we've made recently. I'm a developer in the UniProt team. I'm also, as I said, accompanied by Kate, which is a curator, who is a curator in the team, and Swati, who is another developer in the team. So I have slides here, but this is for support really. I'm just gonna jump directly in the website just to show you a demo directly. So this is the UniProt website, the homepage that you can find by either Googling UniProt on, you can search for UniProt on Google or any search engine, or you can type uniprot.org. You will find this homepage. In there, this is the entry point into all the data that we have in UniProt. Just before really diving into the website, just to say that UniProt is a consortium, and there's a lot of people working. So there's Kate, there's Swati, there's me, and there's all of these other people on the page at the moment. They're split into 3 different sites. So the EBI in the UK, PIR in the US, and SIB in Switzerland. So what is UniProt? UniProt is a comprehensive, high quality and freely accessible resource of protein sequence and functional information. So it contains primary sequences, including sequences of isoforms also, physiological protein functions that includes subcellular location, pathways, reactions, interactions, involvement in disease, and plenty of other functional notations, structural information that includes topology and also access to the 3D structures directly from the website, and some data analysis tools such as BLAST for sequence comparison and multiple sequence alignment. As I said before, this is a resource provided in collaboration between EMBL-EBI, PIR, and SIB. So what does the UniProt website do? So it aims at allowing the access to all the data that we have that we are collecting through the UniProt consortium, and also meet the evolving needs of our users. So we really need the users' feedback to be able to develop the website and improve it, and provide all the data that might be of use to users. So that includes cross-references, catalytic activity, proteomes, functional notation, structures, all of the things that you can see on the slide right now. So what are the core principles of the UniProt website? So first thing is to find the information that you want. So there's there's an advanced search available on the website. So you can search by plain text or by searching for specific fields in the data that you want to look at. Also batch retrieval of data from UniProt website. And all of this it's flexible through filtering. You can also customize your views. So you can customize the view on the website and view only the bit of information that you're interested in. Or you can also customize the download options that you will use to download that data and do work with the data you will retrieve from the website. You can explore all the proteins entries with comments, feature, data provenance, and proteome and sequence cluster collections. With that, you will be able to analyze with tools like BLAST, Align, Peptide Search, and ID mapping that will be integrated in the website. All of this information can be visualized. So some of it is plain text, but some of it can be visualized into specific visualization that I will show in a bit. And finally, you can download all of this information. All of the information that you will see on the website, there will be a way to download it. It's just a matter of exploring the website, and you will see the different download options in multiple formats or different options of customization. So again, this is the homepage. So I will just jump to the real website and show you what it is. So here in the main part of the website, this will be the main entry point into the data. Here you can search for anything. This will search by default in UniProt KB. So here there's a bit of distinction to know. UniProt is the website, and it's a consortium. It's all of us that are providing this data. And UniProt KB is the main data set of UniProt. So it stands for UniProt Knowledge Base. And this will be most of the time what people refer to when they say they're looking into a UniProt protein or UniProt entry. We also have proteome information, protein clusters, which are called UniRef, and also sequence archive, which is called UniPark. On the homepage, you also have access to different tools that we provide at the top here, and also information that can be quite important to the current release that you're looking at, because we have regular releases through the year. And so this is the 4th release of 2023. And it's quite important if you want to compare or reproduce experiments to know what release your data is based on. Also, you will have the help link here that will point to the help center, and also a contact form here to reach us. Scrolling down, you can see that there's also some other supporting data, which is not the core data of UniProt, but still available if you want to search or access the data through these entry points or through keywords, taxonomy, for example. Some blog articles that we have related to the data, information about the latest data releases here. The tools are also linked here. So all of the 4 main tools that we have on the website are available here. Some information about help and trainings, and some information about the data itself. So how to download the data, different ways of downloading the data, and parametric access. All of this is documented in the website. Okay, so now I will look for proteins. So let's say, for example, that I'm interested in the APP gene. So I will look for APP. I will get a list of results. So if it's the first time that you use the website, what you will see is a pop-up that will tell you in what way you want to view the data. So here we have a table of data that might be overwhelming if you just want to explore really quickly the data, or if you don't want to see that much. So you will have the option to view by card, which is a bit more condensed and overview view of the results you have there. And here you can see the same list of proteins, but not in the table mode. Okay. Going back to the table. So here I've looked for APP, but I've looked for APP as plain text. So really that could be returned from the gene field, but that could be returned from anywhere. And really what I'm interested in here at the moment, just for this hypothetical case, is really the gene. So what I could do is go to the advanced search. Advanced search, and then I will see that here I have a way to search for any of those fields. So there's quite a lot of ways to search. And so here I know that I want to search for gene name. So I will look for gene name. And here I will enter APP. What I will do is I will do this advanced search. So you can see there's a bit of difference. Here there's 200,000 results. But if I'm really looking only for gene names, there will be 5,415 results. Also, I mean, there's a suggestion here to refine your search directly from here. So here I could also have clicked directly for gene name, and it would have replaced the search to say, okay, search for APP, but only for genes. As you can see, that can be present in other fields that might be of interest to you or might not be of interest to you. Same thing, I could have looked for APP human. And so I would have had the human one. But if I really wanted to refine and not do a plain text search, but really look for that, I can combine those search, and I will say, okay, gene name, I want that to be APP. And then I will add a field, which is the taxonomy in that case. And I would want the taxonomy to be only for human. And so here I could have entered directly the taxon ID if I know it, but I can look for human. What you can also look for is taxonomy at a higher level. So I could have looked for primates only. And so here you see there are only 11 results. What you can see here is the first one is gold here. So this shows that this is part of the SwissProt dataset or reviewed dataset. These are entries that have been reviewed by a curator, just like Kate, for example, that assess that the annotation in it are correct, and curators have been adding this data, and all of those are part of the unreviewed set. Doesn't mean they're bad, no. It just means that no curator has annotated this. So all the annotations come from other methods. That would be automatic method. Okay. Let me just check. So what else can we show here? Advanced search. This is available here. You could also have reached the advanced search directly from the page here. On the side, you can see, so I'm just going to expand a bit the search so that it's a bit more useful. You can see here, there are some quick search on the side with count. So you can see that if I restrict by only the reviewed one, so I had removed the human filter here. If I restrict only by the reviewed one, I will get 24 results, and you will have all of this data here. Similarly, I could have restricted by other characteristic like the protein existence or a protein with a 3D structure, and then that would give me this result set. All of these filtering options here are available in the advanced search also, so that could be alternative ways of doing the same thing. Okay. What you could also do is search for everything. So here, sorry, I'm going to go back to the home page. If I were to click here on the proteins card here, I will end up on that overview of UniProt KB, explaining a bit what it is, and I can start searching for all the data in UniProt KB. So you can see that in UniProt KB, I have that many entries, and they are split between reviewed and unreviewed, as I've explained before. But what you could also do is, let's say I'm looking only at the reviewed one. Something that's been added recently in the new website is that group by option. So you can say, okay, in this set of 500,000 plus proteins, I want to see how they split by taxonomy. So in there, you can take a bit more time because it's quite computationally intensive. It will be, those entries will be grouped by taxonomy, and then I can explore them. So, okay, I have 3% of them that are from viruses, and the rest are from cell organisms. And then I can dig into those and dig through the tree of data. So here, you only see the proportion at the top level. But if I want to jump directly to the second level, so to, let's say, I'm going to go in eukaryota. So I will click there, and it will use eukaryota as that top level. And then you can see the split between eukaryota of this search. And that could work also for any search that you do. So if you do that plain text search for APP, you can also do the group by taxonomy. And then you will have a different numbers in there, and that you can still explore. Here, if I were to go up to human, for example, that would be a bit deep in there, and it would take a bit of time, but I can just directly jump to human by searching for that. Or I could jump to primates. That's going to be a bit more interesting to see a split. So primates. And then you can see that this is the split in primate, and you can explore the data this way. So this is through taxonomy. And if you're interested, so okay, now I see that there are 1,464 results for APP in Hominidae. So I can click there, and I will go directly to that search for APP in this taxonomy. Similarly, there's you can do the same thing for here for keywords, gene ontology, and enzyme class. You can explore that tree of data this way. This is quite new on that new website. Now I've seen the result set that I like. So what I can do is download it. I can just click on download, and I will have the option to download everything in there in different format: FASTA, TSV, Excel, JSON, text, all of that. FASTA is just going to give you the sequences. But if I go for TSV, tab separated values, I can start customizing the result in the same way. Sorry, I can start customizing the results in there. So I will get columns of information for reviewed and tree name proteins, name, gene date, gene names, and all of that. And I can add more information. So if I look for domains, for example, let's say this one, I can add it, and that will be added in there. And if I want to have a preview of what the data will look like, I can click on this preview button, and then you will see that the file that will contain, I don't know how many, 1,000 plus proteins will look like this. Okay, this is what I want. Okay, then I can download it. You can have also, you have here a quick way to generate a URL so that you can use for priority access. So at that URL, you will be able to download the same data, and that could be used for any pipeline that you have in R, in Python, in whatever language that you're developing in. So you can customize the columns here in your download, but you can also do that here when browsing. So here in the same way, I can say, okay, show me the domain information. I select that, I save, and then you will have the domain information added in there. So you can do that with a lot of information. I don't recommend to check everything, to tick everything, because that's quite a lot. But anything that you might be interested in, there is a column for that, and you can look for that and add it in there. Okay, I'm just going to be set to the default set of columns. Here, if I go back to everything, you can see there's 250 million entries in there. That's quite a lot. I was still able to download it in there. So this is something that will be actually too big for the browser to be able to download. And so we don't actually recommend, well, we will not allow to download it directly. What you will need to do is generate a file, and this will be a file would be created for you by the website, and it will be sent to you afterwards. So here in this case, I can submit this. This is a job that will be run on our servers, and whenever this file has been generated, then you will be able to download it after. The good thing with that is you can still have access to all the customization options that I said before. So you can still change all the columns and all that for those millions of entries. It will take a bit of time, but you will eventually get it. This is maybe not a good example, because what you should be doing if you download everything is go to our FTP website. So this is documented on the help page, and just download the pre-generated files from there. So you can download the set of everything in UniProt KB directly from FTP. And so this is available in the help. But if you wanted to have customization or big subset of UniProt KB, this would be a way to do it. Okay, let's jump into a entry. So I would use that one. So the APP, well, which one I want? I want this one. So this P05067 is fairly well annotated, and so that's going to be a good example to go through the different sections. So here I jump to the entry view. You can see that at the top you have the different function. You have access to the different sections here of the entry, and so if you're interested on a structure, you could click here. That would jump directly there. But yeah, I will just scroll through the page and show you what you have. So these are function annotations that have been added, and if you want to know where they're coming from, you can see that in there you have references to permit IDs, and also these tags that are sprinkled a bit in the page will tell you where all of this information is coming from. So all this paragraph is coming from these 3 publications that are listed in there. You can view the abstract in there, or you can go to the PubMed website or another website to have a look at this publication. And you can see that it tells you also what type of annotations you have, what type of evidence you have for this. So this one is a manual assertion based on experiments, but you will see going through the data, you will have different types. So that way you can assess if you're, where that annotation is coming from, if it's been manually added by a curator, or if it's been automatically added by some kind of pipeline. So I'm going to close this and then scroll through. So here, still in the function section, I will see this visualization that represents the protein sequence. So it's a sequence, so it's a protein that has 770 amino acids. And here I will have some annotations. So that first one in there, this is this binding site. Then I have another binding site in there. So, I mean, it's a bit big, so I can restrict or zoom in to the sequence by selecting this side of the viewer, and then just scroll through the sequence. So you can see, okay, these ones are not in view anymore, so that's why they get it out. But if I go back in there, you can see that this first binding site corresponds to this sequence in there. Similarly, as before, you also have the information about where this annotation is coming from. So this is from a publication. These ones are from 2 publications, also a combination of other sources and a rule annotation. And so you will see this kind of visualization throughout the page. So you also have co-annotations, keywords that are related to that proteins, a lot of links. So if you want to follow up on that and have more information about those keywords, you can click through them and it will take you to a specific page. And you can see the cross-references where on other resources, other websites that can be interested to follow if you're interested in this kind of, for example, here, enzyme and pathway databases. Different names and taxonomy, so recommend these names of the protein and gene names. The taxonomy that you have here, you can see the whole lineage in there. Maybe, so subcellular location, you have a visualization of where this protein is in the subcellular locations. Disease and variants, a bit lower than the page, so variant data that you have available in the website with the different consequences. So here APP is really fairly well annotated because it's related to Alzheimer's, I think. Yes. Sorry, I'm not a creator, I'm a developer, but yeah, this one is linked to Alzheimer's, so that's why it's a good example because it's been well annotated, and I can show you a lot of data. Maybe I can also show you the structure. So if you go to the structure section, you will have access to all the structures that are related to that protein. So you have PDB structure, but you also have on most of the import KB entries, you have access to AlphaFold. So in some cases where you don't have PDB, you will still have AlphaFold available, and this data is integrated in the website, and you can see it in there also. Okay, family and domains. Similarly, you have this mini view that is focused on helix. Sorry, not family and domains. I'm still in structure. But here, this is focused on helix, beta strand, and turn. So you can see where in the sequence you have these features. And then if I jump to family and domain, it's a bit lower. Then you can see regions, domains, and all of that. So if you're interested in a specific type, you can also restrict to motif. So here you have this motif. I'm not really sure what that is on this view. So what I can do is click on it and it will highlight it. And I can see it's right in the middle. And then sequence and isoform. So in this case, there's a canonical sequence for P05067 for the APP, for this APP protein will be this one, but it's also associated with isoform, which are kind of variants of that same proteins that have some differences from the canonical one, and you can still access the sequence here. So you see this one is shorter, and all of these are listed in there. And finally, what you can see is also at the bottom of the page, similar proteins. So similar proteins are other proteins in our database that either have 100% identity, 90% identity, or 50% identity. It's a quick way to have already pre-generated set of proteins that are similar, and you can see that, for example, in this one, there is another protein in macaque that is 100% that has 100% identity with the one that we're currently looking at for human. And if I want to expand the set, I can jump to 90% identity, and you will have a lot of different organisms that have 90% similar. And you can see that they actually have the same name in this case, and you can browse all of them. So these sets are what are called UniRef that we've shown in the home page. I can show again. These UniRef clusters of identity at 100%, 90%, 50% identity. You can browse them through there also, or you can search for them in there. And if I go back to the sequences, you can see that all the sequences have also a unique reference to UniPark. So UniPark is the dataset in UniProt of unique sequences. So you can see that if I jump to that UniPark entry, you will have information about the sequence and about all the cross-references, so all the different entries where it's found, and also the cross-references that I might have to over databases. Okay, back to this entry. Let's go back to the top. So here, this is the entry view, which is data from UniProt. But okay, we can see the different tabs that you have here. So you have history. So entries can change, and so you can still have access to the previous versions in this tab. So annotations can be added, the canonical sequence can be changed, but we keep track of all of that, and you can still have access to this protein as it was, for example, in this case, in 2021 in our database. And you can scroll back you can go back in time until the first appearance of this entry in the UniProt database. You also have a tab with just the external links. So all the external links related to this entry will be found here. PTM database, sequence databases, genome annotation databases, and all of that, 3D structures. All of this will be listed here without the plain text that you have on the entry and without the visualizations. You have also all the publications that this entry where you can find a reference to this entry. So you can see there's quite a lot, and you can still filter through them. So, for example, there are 1,359 publications that refer to the function of this entry, and so you can explore them in there. What you have seen in the entry is also these mini views with a bit of for example, the domain annotation, the structure annotations. They're a bit spread throughout the page. And if you want to compare, okay, I have domain here, but I want to see if there's a variation at the same place, what you can do is open the variant viewer. The feature viewer. The feature viewer will show all of the different visualization in there. So this one is a bit overwhelming because there's a lot of annotations, but still that can let you compare. And within the structure, so from the first amino acid to the last amino acid, where are the different annotations? So here you have structural annotation. So you can see this is an helix, and this helix is right in the middle of this region, which is a CUBD domain. I'm not really sure what it does, but yeah, this is right in the middle of this region. You can have a site. You can see this is on this specific peptide. And so you can view all of this together. Similarly, you can, if you scroll a bit down, so here I've selected a binding. And if I scroll, so this is not a good example here. I will use the AlphaFold one because it has the whole protein in there. So you can see that here I've selected a domain before, and it will highlight it in the AlphaFold structure. But if I select this peptide, it will show it to me in the 3D structure, in the AlphaFold structure, and I can see these things in context. What I can also show is something quite new, is the variant viewer. In the variant viewer, you will see variants annotated either by UniProt directly, so you can filter them here, and you can see that there are variants here, but also imported from other sources like CleanVar or other large-scale studies. And they are categorized by if they are pathogenic or benign, or if they have other things. So you can still explore them in the table. And similarly, if you click on them, you will see them highlighted in this view. And you can still interact with the view to be able to see where in the sequence this variant can exist. Okay, that's the entry view. So now if I talk about the tools that you have access to. So here, for example, so let's say that I go take this sequence in there. I will just copy it. And I will want to blast a sequence that comes from my experiment. And so this is not really this sequence, but this is something a bit different. So I will just modify it a bit. This is not what you would do, but yeah. TR, something like that, and we remove this bit. So this is not the same entry anymore, but I will still run the blast against it. So I will try to blast it. So what I can show you is that, so when you run a blast, it is a sequence similarity search, And so you need to define against which database you want to trigger that search. So in that case, by default, it's using UniProt, KB, RefProt, reference proteomes, and SwissProt. So this is the reviewed one and the unreviewed one that are from a reference proteome. You can also expand your search to all of UniProt KB. It's always a trade-off because if the database that you search against is bigger, it will take longer to return. So this one is usually a good trade-off to find good information, but still run in a decent amount of time. But if you want, you can use that one that will have more proteins, but it might take longer. You can also restrict for only UniProt KB with 3D structures, UniProt KB from reference proteomes. And you can also search against the UniRef databases that I've talked about earlier, or the UniPark one. UniPark is going to be quite huge, so it will take a long time to search against that, but you will have all the sequences in UniProt, in the UniProt databases. Down there, you can also customize the BLAST parameters. So I would not recommend to change that unless you know what you're doing. This is not really part of this webinar. I will not go into too much detail into those parameters. What you can also do in there is directly restrict by taxonomy. So if you want to reduce a bit the size of the database you're searching against, you can say, okay, I'm going to search only in primates. So you can do that. Or you can also, I can remove that and search only in human. So this is something I can do. You can also name your BLAST. So my last job. And what you can do, what you will do is then later on, you will be able to find it. So when I run this BLAST, I will end up in this view, which is my tools result dashboard, which will list all the different jobs that I've been running. So this is the one I've run just before. This is the one I've just named, my BLAST job. And you will have a bit of information about what type of job that is. In this case, this is a BLAST. So you can see it's taking a bit of time to run. It's normal. It can take a few minutes. I've already run a BLAST before, so this one completed, and I can open it. So here I have 250 results that are found in UniProt KB with my input sequence. So here you can browse the result set. We will have information about how about the identity, the score, and the e-value of the hit. So in that case, it's really similar to the isoform 4 of P05067. And if I want to see exactly what are the differences compared to from my input query to this result, I can click on here, and it will pull that pairwise alignment view, which is similar to the domain visualization that we saw before. And then I can browse through the hit to see what are the differences. So you can see here I had edited this bit of the sequence to remove that, so it picked it up. And you can see that in there. You can also zoom out to have a bigger view of that, but that would correspond to this. Okay, I can also, there's a lot of results in there, so I might not be interested in all of them. So I can still filter in the same way I filter in the search in there. So I can filter only for the reviewed proteins. And here you can see that I will only see the hits that are from the UniProt KB reviewed set. Similarly, here, those histograms, taking a bit of time, sorry. So these histograms are also a way to filter. So you can filter from these hits only for the better scores. So let's say I will select this, and it will only reduce, sorry, it will show the data only for these high scoring hits. Okay. A good thing about this dashboard is that your job keeps running in the background. So you can see that I opened this BLAST job, and in the meantime, these two jobs completed. So it means that you can still browse the website while it's running. Your job will not be lost. And you can see here that since I was browsing the website, I also got these notifications at the bottom of the page that told me that those jobs are finished if I wanted to have a look at them. So I will discard them because I've seen them. And so you can have a look here at the dashboard. You can view that through here directly. or if you click there, you will have the full view dashboard with all the jobs. These are jobs I've run before with other parameters and all of that. So these jobs will be available for 7 days. You will still have access to the results for 7 days. What you can do after 7 days, they will be kept for an additional 7 days here. So the job results will not be available, but at least you will still be able to resubmit them. So it will have kept input parameters, and you can still submit them. If you're not interested in them anymore, you can delete them also. But eventually, they will be automatically deleted after 14 days in total. And they will not be deleted if you star them. If you click on the star, it will be kept for as long as you want. What it can be useful for is that you can run a blast against a specific release. As I've shown at the beginning, you have this release here, 2023-04. So I've run a blast. And then actually I want to run it again whenever the new set of results will be published. So that will be, I think, in November. So then I can go back in November in this dashboard and run this job again, because it will not be kept in there. So this is something that will be kept in your browser in this specific computer that you're using. So if I'm using Chrome, but if afterwards I'm using Firefox, that will not keep the result. We don't have any login system at that point. So it will only be kept on the same browser as long as you don't clean the cookies, or you don't clear the cookies or anything like that. And so if you're using another computer or on your phone, for example, that will not be kept. In the same way, we're also keeping the search customization that I've shown before. So when you are doing a search and you customize the columns or customize the view, that will be kept in the same way. And if you clear the cookies, then that will be reset. Okay, back to my blast job here. So here I can see that this one is, so my input query, the one that I modified, is really similar to this and really similar to these others. So what I might want to do is actually align them just to see how different they are or how similar they are. So here I can see pairwise alignment between my input query and this hit. But what I can do is also align against multiple ones. So here I've selected those two because they might be interesting to show alignment for. And so here I can have access to the different tools. And so align is one of them. And you see that I can align the selected ones with my query. So those two with the input query. So it will align 3 sequences. What I end up in here is the aligned form. So here, before I had the blast form, now I have the aligned form. So let me just go back to the blast to select them again. So this is the way to submit alignment jobs. So this is using a cluster omega as an alignment algorithm. And so here you will see that I have this sequence, which is the one that I've used as an input for blast, which is the modify one. I have this one, which is P05067 from SP from SwissProt from the reviewed set, and this other one. Okay, and then I can align them. And similarly, I will end up in the dashboard. So same things that will be kept for 7 days. The parameters will be kept for a total of 14 days, and you can keep that forever if you want by doing the star. Alignment usually is a bit quicker than blast. So hopefully that should run. Otherwise, I have backup ones I've run earlier. Okay, this completed. So since I'm already in the dashboard, I didn't get the notification because I'm already looking at that. So now if I enter this alignment view, it's similar to what we had before at the bottom of the blast page for the pairwise alignment, but in this case, this is multiple sequence alignment. So here I have 3 of them, and I can see the 3 sequences, the input from before, and those 2 hits from the previous blast. There are also 2 views in there. So you have the wrap view, which is the sequences, which is similar to the raw output of the tools, of the align tool, the cluster omega tool. But I can also have another view. So this is a zoomed out view with all the sequences, and you can easily see the gaps and the differences in there. So I can just scroll to the bit that is different if I'm interested in that. You can see by default it's coloring by similarity or conservation. So yeah, this is a default view. I can choose to color through another color scheme. So anyone that is used to the cluster tool might be used to this color scheme. So you can customize it in the same way, or you can use any other color scheme. By default, it's similarity. And what you can show also in the sequences in there are annotations. So same annotations that we could see before on the entry pages, you can compare them here in alignment. So you can see that here it's selected by default signals, so it's highlighting signals within these proteins. You can see there also that these two sequences have this signal here. So if I click on there, I have more information about that. And one interesting use of align then is if you have a sequence that you don't know the the function of, then you can compare them with other proteins that we have in the data set that we do know the function of. And then you can assess if by similarity, if this part of the sequence is conserved or not. So here in this case, it's conserved, so it's quite likely that you will have the same signal in this hypothetical protein. And if I'm interested in another type of annotation, so here I will get the annotations from this A4 human, and I will look at domains, for example, and you can see that all of these are in conserved areas. So I'm not sure if I've touched anything which is not conserved, but for example, if there was a domain here in this human one, and there would be a deletion here, I could think that maybe this domain is not conserved in this protein. Okay, other tools that we have access to is also peptide search. So BLAST will be a tool to look for a sequence by similarity. Peptide search is a bit similar, but it will allow only to look for exact sequences. So here, if I look for this specific sequence, it will not find the similar ones. It will find the exactly equal sequences. So any protein that contains this sequence will be presented in the result set. So it might take a while to run. I don't actually know. I haven't run one before. It's fine. Basically, it will show you a result set from UniProt KB. And so it's quite similar to what you would see here. You can also customize the view by filtering within the result set or look for anything within this result set. And the last one is ID mapping. ID mapping, you will have a list of IDs and you can map them to UniProt KB or from UniProt KB. So say that I have a list of UniProt accessions, I can paste that here and then do the mapping, and then I will get a full view with all the results customizable and all of that. What's interesting also is that you can also map to and from other databases. So say that you have a list of PDB accessions, you can paste in there, you select input as PDB, and then you map to UniProt KB. So you can find all the UniProt KBs that map to a specific set of PDB accessions. Same way around, if you have a list of UniProt KB, you can map them to PDB, for example. What you cannot do, because this is UniProt, so basically the center of this mapping tool is UniProt, you cannot map, say, PDB to EMBL directly. You will have to jump through the UniProt step. So you will have to have UniProt somewhere, either as a from or as a to of this search. So here I have run one before. So this one completed. So if I click on the resubmit button, I will see the input I've used. Oh, sorry, in there. So if I click on the resubmit button, you can see I have a list of accessions in there. Quite a lot, actually. So you have, this many accessions. So if I open the results, you will see that it mapped 1,449 IDs to these entries. So it found that it was this entry. And so then you can, just from the accessions as an input, you can still get all the columns that we have in the UniProt data. So here, if I look for domains, similarly, then I can display the domains for this list of input that I've used. Okay, I will just check that I have covered everything I wanted to cover for tools. Okay, this is fine. I will finish up the presentation. So we'll jump through those because it was back up. So all these slides will be shared with you a bit later. So you can go back to it, and everything that I've explained now should be explained there also. So we also need your help to improve the website. So anytime you are in the website, you will have this small tab on the side, which is a feedback. So you can open it, submit a quick feedback. This is actually really quick. So you can just rate your experience from this on this scale, but then you can also enter a comment. So that can be really helpful if you say that, okay, this bit of the page doesn't work. This is this can be improved or whatever. We look at all the feedback that you submit. I will skip. So if you need further help and documentation, as I said before, you have the help at the top of the page, or you can contact us. Contact us. You can ask for anything. We program about the website, program in the data. You see that you have a publication that is referencing this protein, but this is not on the UniProt website. You can contact us, and we will look into adding that. As I showed before, there's a huge number of people working on UniProt throughout the three sites of the consortium. So someone will be able to help you on any kind of topic that you want to raise or you want to ask about. Okay, as I said at the beginning, you have access to the help straight from the homepage in the need help section. And this is a list of UniProt related webinars that will be happening soon. So the first one, an introduction to UniProt, is the only one that at the moment you can register to on the EBI training website. So if you want to have another introduction to the UniProt data, you can register already. Then later on, you will have in November accessing UniProt with the API via the API using Python. So I will be giving this this webinar that will be accessing, viewing all of the things that we've viewed today, but through programmatic access. And then you will have the other webinars that are listed on this page. This is the funding for UniProt. So this is quite important for us to be able to do the work that we're doing. So this is this. Then regarding the EMBL-EBI webinars, you can browse the EBI training website, and you'll see that the upcoming webinar in this series is molecular building blocks of life. So you can access to it. You can access it directly to the EBI website. So now I will open the floor for questions. Back in the view. So Swati, have we received any questions?
Yes. The first question is how UniProt Blast is different from NCBI Blast.
Yeah, so UniProt Blast is actually, so UniProt Blast is running at EBI, but it's using NCBI Blast. So if you can see, I will open a Blast job here. So you can see in these tabs, you have related information about the Blast, and you have the different input parameters, and you can see that we've used the NCBI Blast tool in our servers with these specific parameters. So if you want to reproduce that, you can do that, and you can have access to the raw output. And you can see this is using this is the raw output from the NCBI Blast tool. So yes, this is technically the same. The advantage of that is that it's integrated into the data. You have filtering of results in there in the UniProt website, and you can jump back and forth in the results. Yeah, you can do directly the entry that corresponds to that. So it's yeah, really the integration into the UniProt website. Okay.
Okay. The second question is I've answered, but it would be better to show it in the demo, I think. Is there a way to put a list of proteins in UniProt and then download the information for those proteins?
Yeah. So a list of proteins, it depends on what you have as an input, really. If this is yeah, from external accessions are not UniProt, they might be listed. So they would be using ID mapping in general. So if this is UniProt KB, a list of UniProt KB accessions, you can list them here, just as I've shown before. Sorry, up. With that big example. So here I have a list of accessions. What you can also do is a list of gene names that would be one of the inputs in there. So you can search for gene name. So if I do APP and UniProt KB, then I will get a list of let's see, but yeah, it will get a list of UniProt KB entries that have this gene name. So I think that covers it, Swati. If we still have a few minutes, I can show a bit more of the past results, of the tool results. And what I wanted to show is also that we have those tabs that that can be a bit useful to explore the result of BLAST. You can see the taxonomy distribution within the BLAST result in there, the hit distribution, identity or score, as I showed just before, the text output, the raw output from the tool, and the input parameter, the API request. So if you wanted to do that to access the EBI service that would do this, you can reproduce that with curl using this. Similarly, for all the tools, we have these last tabs are always the same. So you have always the raw output of the tool. So in that case, that would be the raw output of Crystal Omega. The input parameter that we use, so that you can reproduce that even if you have that running locally or on your own service, and the API request if you want to query the EBI service. And here on top of the alignment, you also have the phylogenetic tree available. The percent identity matrix, in that case, is quite similar, so it's not really interesting. So the identity matrix of all the proteins aligned. Yes.